Okay, I think we can get started on the session on mycoheterotrophic plants today. So welcome to the session. Really looking forward to a lot of great talks today about these fascinating plants. And Tom Gidnish mentioned some of them this morning. So first of all, I'd like to thank our funders and co-authors, several in the room. I'm Sean Graham. I'm not Craig Barrett. Craig is sitting there. So we decided that I would give this talk today. Several other people on the author list are here, including John Freudenstein and Hana. Two of my students, Izai and Philippa, who will be talking later on today. Actually, Izai is talking right after me. Philippa is going to be talking at the end of the day about new carnivorous monocot lineage. Vincent Merckx is not here. Vincent Merckx from Naturalis, and he made a lot of this work possible. So first of all, I'd like to just briefly thank the organizers, students, and everyone who's working very hard to make this already a terrific conference. So gracias. So my talk today is divided into three parts. I have a short introduction on mycoheterotrophic monocots, and then I'm going to spend the bulk of the time splitting it into two pieces, thinking about the phylogenetic placements of these taxa, and how we can refine our knowledge on that using plastid genome data. And then I'm going to finish off by talking a little bit about the natural history of genomes. So we often use that term natural history to think about organisms. Well, genomes are part of organisms too, and so we can also think about their natural history. So this is a little bit of an introduction to the ecology of mycoheterotrophs. So plants have these amazing relationships with fungi. Only about 500 species of plants are full-on mycoheterotrophs. This means that they've lost photosynthesis and they fully rely on soil fungi for their fixed carbon needs. But of course, 85% of plants have some kind of mutualistic relationship with fungi. And so there's this ecological spectrum of relationships between plants and fungi that ranges from fungal parasitism through to mycoheterotrophy, which you can kind of think of as a kind of parasitism too. Mental note to remember to do this. So thank you, gracias. And thanks to my students for pointing this out. Yeah, so this is the spectrum of relationships, the ecological spectrum of relationships between plants and fungi. Full-on mycoheterotrophs are over here that have lost the ability to photosynthesize. There are also mycoheterotrophs that do photosynthesize for part of their time, but today I'm going to be focusing on the full mycoheterotroph. So only about 500 species of plants do this. This is a phylogeny with mapped on it just there's a button of death here that I must not touch. So this is a phylogeny with mapped on it the number of species of mycoheterotrophs in the different orders of flowering plants. And you can see that the best group of flowering plants, the monocots, right, has most of the mycoheterotrophic lineages represented by the bars, which represent the number of species. And those 500 species are highly dispersed across 8 families of monocots in 5 of the 12 orders. So almost half of the orders of monocots have some kind of full mycoheterotrophy. And there are several families with asterisks here that are full-on mycoheterotrophs. They only have mycoheterotrophic plants. This is an image of some of those plants, and maybe my favorite of all time photograph of mycoheterotrophs. This is, I think, the fingernail of Vincent Merckx. I think that's Vincent. And then this little tiny plant, this wee plant, is in the Triuridaceae. It's Triuris. Amazing. They're amazing, spectacular-looking plants. To this pantheon of families with mycoheterotrophs, we recently added an 8th. Well, it's kind of cheating because we knew about this species. It's obviously fully mycoheterotrophic, but we've split it off as a new family, a new monocot family. So the basis of that is partly phylogenetic and partly based on morphology. There's many morphological differences between Afrothismiaceae up here, and its two most closely related families, Thismiaceae, which is fully mycoheterotrophic, and Tachyaceae, which is photosynthetic. So I won't say much more about that. You can go and look in the paper that recently came out by Martin Cheek and colleagues. So the first major part of my talk, we now have a really good fix on where most of the mycoheterotrophic lineages place in monocot phylogeny. So Tom talked about that a little bit this morning, which was really unknown in many cases, even at the last Monocots conference. So I'll just point to you several of these papers, data from all three plant genomes, but the plastid genome has played and continues to play a really major role in thinking about the phylogenetic history of plants in general, but also mycoheterotrophic lineages. However, it is challenged by several lineages which have very, very long branches. So I would say that we might have reached our limits in what we can do with plastid data. And so I'm going to talk a little bit about that and some alternatives. So those very long branch lineages include multiple ones in Orchidaceae. So Orchidaceae is by far the richest natural history of mycoheterotrophs. There's probably been at least 30 origins in orchids. Dioscorales is probably the next richest clade. So this is the yam order, Dioscorales, which has now 3 families of mycoheterotrophs. And in one of those families, Burmanniaceae, there have probably been around 5 or so origins. So these are by far the richest in terms of numbers of origins. And here's the problem. So I'm going to be showing you several slides with trees that look a bit like this. And I just want to point out a couple of things that kind of clue you into what's going on here. So these obviously have very long branches. These are mycoheterotrophic lineages. They have lost photosynthesis. But I also want you to focus on the backbone. So in this case, this is the rest of monocots, including the very rapidly evolving grasses up here. So look at that little tiny scale bar there. That scale, if you kind of multiply through here, there's probably an average of 2 or 3 substitutions per site in Afrothismiaceae, which is probably one of the longest branch lineages of plants, highly elevated rates. So this is a real challenge for making any kind of phylogenetic inference. So why is it a problem? So Marybel Soto Gomez in my lab did her PhD on this partly. And this is a diagram. She did a lot of different analyses. And this is a diagram that shows what happens with Afrothismiaceae when you include it in an analysis. You kind of breathe in it, and it goes to a different place. It's really unstable in where it places. And so this place is absolutely ridiculous. It's placed within the Poales order, right? That's just ridiculous. It's not a Poales, obviously, but it's playing a role as a wild card. In doing so, this is the same tree up here. So this is this part of the tree down here. So here's grasses and relatives. But you can see that the backbone, these are whole plastid genomes, but the backbone is basically just collapsed in terms of its support. So they tend to destabilize local relationships when you add these very long branch taxa. And then the final problem is that if you have more than one long branch taxon in the analysis, they tend to clump together. This is classic long branch attraction or very long branch attraction. And it's not just a problem for parsimony, it's a problem for model-based methods too. So this is an ML figure of orchids where some very long branch orchids are clumping together in this broader phylogeny of orchids. These are the long branch orchids here, and this is not real. They're in the wrong place, and they're kind of grouping together. So it's a clade in quotation marks. So what can we do about that? Initially, I'm not going to go through this. I don't have time, but I'll leave this as a final slide as a possible discussion point. But I just want to point out a few things. So I think we're maybe at the transition from mycoheterotrophs for the very long branch taxa, maybe partly abandoning plastid phylogenomics and using other genomes. I'm going to focus on the mitochondrial genome today because I think they've been neglected, badly neglected. So just a few points about that. They're a little bit harder to obtain from genome skim data because the concentration per cell is lower. But when you do get them, they tend to have very few missing genes, whereas plastid data often have many missing genes. These genomes are very slow, which is probably why they've been neglected in use in big studies. But in combination, the genes are really good in phylogenomic analysis. In addition to being really slow, which is also an advantage if you have long branch taxa, long branch effects are weaker when you have slower genes overall. The mycoheterotrophic genomes are also rate elevated from mitochondrial genomes, but proportionally not as much. And so I'm going to show you some slides that basically suggest that for these very long branch taxa, we should probably be switching to mitochondrial genomes. So this is an analysis that Craig did of some orchids that include some very long branch taxa. And the tree on the left is his results using mitochondrial data. This is what you were looking at with plastid data. So we have these very long branch taxa grouping together. Look at that enormous substitution rate. This little graph here shows the difference between green photosynthetic taxa and mycoheterotrophs. So they're vastly rate elevated, and look at the scale here, number of substitutions per site. Contrast that with the mitochondrion, which are much slower overall, and they have less of a discrepancy between the rates of mycoheterotrophs and green taxa, even for these very long branch taxa. And so when you use those in phylogenetic analysis, and Craig did some other things here, he took a kind of heterotachy, which is a broad issue that we should probably all be thinking about. But when you do that, the clumped taxa become unclumped, and the lineages go where we think that they belong based on other lines of evidence. So we also use this approach for Dioscorioles. So the tree on the left is from Marybel Soto Gomez's thesis, and you see the sort of classic effect when you include very long branch Afrothismia. This is a cladogram, so there's no branch length here, but it is a very long branch. Basically, it collapses bootstrap support all the way down, and it goes in a place that it actually doesn't really belong within Burmanniaceae. So when we switch to basically the same taxa for mitochondrial genes, we get a very different picture. So the analyses are very stable, so you can do lots of different methods, and it gives you the same answer, and the results are very well supported. And the very long branch taxa, well, they're still long, but they're not as extremely long. So we put more faith, if you like, in mitochondrial evidence for these very, very long branch taxa. So having said that, I think that we are going to continue to use the plastid genome because it does work well if the taxa, if the lineages are not very long branch, but there's also very interesting information about their evolution. And I think this will continue. People will be collecting additional lineages of mycoheterotrophic plants. Here's a kind of selection of a few papers that have come out very recently. So I think this will continue even though plastid genomes might be abandoned for the very long branch taxa for phylogenomic purposes. But it's still interesting to include them because we can tell a lot about their natural history, their evolution. And so the rest of my talk is just a quick walk through some of the natural history of the plastid genome, which is absolutely fascinating. So here's a slide from Craig. These dots here represent different genes in the plastid genome, so roughly 80 protein coding genes. And then these are taxa within a group of orchids. This is all orchids or just a group? I can't read that. It's all orchids, no. So the dark squares there represent when there is a gene retained, and the white spaces are when a gene has been lost. So you can see there's a range of situations here, and we can use information like this to make predictions about patterns, to go from pattern to process, to make predictions about how genes are lost. This particular taxon here is a recently sequenced one right here, and you can see that it's missing most of its photosynthesis, ATP synthesis genes, and it's also missing some of the plastid apparatus for continuing to be a plastid. So more than 10 years ago, Craig and colleagues came up with this scheme for thinking about degradation of the plastid genome. This happens when you lose photosynthesis. You lose most photosynthesis genes, and eventually only a few non-bioenergetic genes are retained, and things like the ribosomal apparatus. and the loss might start before the loss of photosynthesis, and it might end up in the loss of the plastome, but we've never seen this in mycoheterotrophs to date. So as we continue to collect more plastid genomes, we can refine our understanding of what's going on. And so I think we're sort of at a sort of more probabilistic level, or approaching that of being able to say, what are the orders of gene complexes that can be lost, and what is the variation in that across taxa? And so this 7 years ago paper tried to capture some of our expanded knowledge of that. So it's also useful to go into individual groups and to look, if you have a phylogeny, it's a good reason to study them, to get a phylogeny. If you have a phylogeny, to map the gene losses onto phylogenies. And so these two studies from Craig's lab look at gene loss in the plastid genome of relatively recently evolved groups. And you can, if you look carefully, you can see that there are several lineages that are photosynthetic still. So there's multiple losses within these trees. So contrast that with a similar study, but on Afrothismiaceae, which is probably an older group of mycoheterotrophs. And in this particular case, all of the stuff that happened that you saw in the previous slides probably happened somewhere along the stem lineage of Afrothismiaceae. So this alpha here is 82 genes lost. I think that's actually slightly wrong. Oh no, this includes tRNA genes, so that is correct. So most of what happened here happened a long time ago, and what we're looking at today is sporadic loss in fully mycoheterotrophic lineages that have already lost most of their genes. So phylogenies can be useful for interpreting these kinds of results. But of course, we need to continue adding taxa. So there's a talk this afternoon by Petra Svetlakova on oxygyne, which is an important missing lineage within Afrothismiaceae and a beautiful plant. And then this recently described group within Afrothismiaceae, Relictothismia, which is a paper that basically came out last week. So a new genus of Afrothismiaceae. So really excited to think about those. My last few slides are about a couple of single points, so basically one slide each. We can also use phylogenetic methods to think about this. So this figure is plastid genomes that have been unfolded and laid out, and the lines connecting them represent rearrangements, so inversions, compactification of the genomes. So this is very nice to look at, but it's sort of very hard to interpret in an evolutionary sense. And so what we'd like to do is map those changes onto phylogeny. And here's a study from 2013 that did that. This study is hand-curated, so there's no automatic program that will do this. I wish there were. So one of the students in my lab is working on this problem, Philippa. The final major point I wanted to make is about purifying selection. So when you look at the genes that are retained in plastid genomes, they're retained because they're probably under strong purifying selection. But in looking at these genomes, you always hope that you're going to find ones that are about to be lost. We tend not to see those, and we think that's probably because they're lost pretty rapidly. But you can go in and measure changes in purifying selection, and so this just shows for different tax species in Afrothismiaceae, the bold boxed ones are ones that have decreased purifying selection or increased purifying selection. So it's pretty sporadic. There's no real pattern here that we can see. And when we look in other lineages where this has also been done, the changes in purifying selection are also a bit sporadic. So maybe it's a problem of noise to signal here. We might just need to study more lineages to find those patterns. This is a study, this is kind of my last data slide. A recent study, unpublished study by Craig. So you can use coevolve, which is a method to look for covariation, or uses covariation thinking to look for correlations between things like substitution rates and structural change. So you could ask, are more rapid genomes more likely to have structural change? There's probably a mixed answer there. And so the bars here represent correlations between those things. So for example, this is structural change, the DCJ parameter, and this is DNDS. These are the substitution rate parameters. So this is exciting, and I was going to bring Craig up to the stage to talk about it, but we're out of time. So these are the conclusions. I'll basically leave this on, and I have a couple of slides also for discussion if we have a little bit of time for that. So I think the next person, if you want to go up and get your microphone on, and we have roughly 4 minutes for
Thank
questions.
you so much. So now we have time for one of the questions. Questions? Any questions?
If not, I'll continue my talk. I'm kidding.
Yeah.
So the slide when you were showing Afrothismiaceae with the bulk of plastome degradation happening kind of early on in the evolution of that group, that's followed by species divergence events, and these mycoheterotrophs often get called evolutionary dead ends, and so there's diversification after this.
I know.
Even when they have miniaturized plastomes.
Yeah.
So.
It happens. I think the numbers probably tell the story because there are only 500 species, and so what probably happens is that mycoheterotrophy arises pretty regularly in plant evolution, and there are many ghost lineages out there that have just gone extinct. But there are modern groups that are long-lived. So Afrothismiaceae is probably one of them. We don't quite know its age. There's some discrepancy in the ages. But Triuridaceae is another brilliant example of that. It's a really old family, probably. The crown age is probably pretty old. And there's lots of species diversity in there. So I think on the whole, probably they might tend to go extinct rapidly. I don't like to use the term dead-end species, but maybe it's justified. But there are lineages that have been around for a long time. So what explains that? I don't know.
Thank you. One more question?
We talked about this recently, Sean, but we really need to have ages for these different groups. We need ages for the mycoheterotrophs to, in fact, test the hypothesis that their average stem ages are younger than green plants are. In other words, that would support the evolutionary dead end while still allowing for diversification. And can you comment on what we know and don't know?
I think what we don't know is most of what we don't know. The known unknowns and the unknown unknowns. Yeah, those. I dislike bringing up Donald Rumsfeld, but that's kind of a good thing. So I would basically say, yes, it would be lovely to have those, but there is a major challenge because some of these lineages include long branch taxa that are challenging enough, and some include very long branch taxa. So my perception is that our current methods for dealing with that, Bayesian methods, which are supposed to deal with diversity of redshifts and so on, are basically inadequate to the job. And I think, at least in some of the lineages, and I think this probably explains the massive discrepancies that we've got in, for example, the age of Afrothismiaceae. So I agree, it's worth doing, but I just worry that it's very, very challenging with fully mycoheterotrophic lineages. And in fact, we commented in a paper on using fossil evidence for dating monocots in Botje Lindshoek a few years ago. So Maria Gandalfos, Selena Smith, Will Isles, and myself. So go look at that paper, and we said some things about Triuridaceae in there. We'd love to do it, but I think it's going to be challenging.
Thank you so much. We don't have more time. Thank you, Sean.
Thank you.